Hello, everyone, and welcome to our webinar on the potential of AI in drug development. My name is Charlotte Guzzo, and I'm one of the co-founder and COO at Cynogenetics, where we are on a mission to accelerate the world's transition to precision medicines through a 360 platform that integrates patient finding, screening, and engagement to reduce inefficiencies in clinical trials. I'm very privileged to welcome three fantastic guest speakers today who will put their experience in the field in perspective to discuss our topic. I'll start with Parker Moss. Parker is the Executive VP of Corporate Development at Exscientia, where he focuses on precision medicine and in particular oncology. Parker, can you tell us a little bit more about your background and your work at Exscientia?
Yeah, absolutely. It's a real pleasure to be here. So I have a little bit of an unusual background. I came from a technology background. I was trained in physics and I worked in telecom for many years. And my life changed rather dramatically in 2012. And that was really precipitated by the diagnosis of my 4-year-old daughter at the time, which stage 4 neuroblastoma, which, if you know the pediatric cancers, is not one of the ones you want. So I left my job in the telecom industry and we moved into Great Ormond Street Hospital, which is the big children's hospital in London. And we actually ended up living on that ward for 5 years. I slept 800 nights on that ward over that 5-year period. And during that time, I became more and more interested in life sciences. I remember reading Bob Weinberg's seminal paper on the hallmarks of cancer and being fascinated with that. I really decided to change my life towards this field. So I started off working in the NHS, understanding clinical data and what the NHS could provide in that space. I was very lucky to then be trained by F-Prime, Fidelity's biotech fund, to really understand the kind of patterns of what makes companies successful in this space. I worked for Genomics England for 4 years, leading commercial partnerships there, but also being one of the leaders in the cancer program. And that was great training for AI drug discovery because I spent really 4 years trying to understand the target space. But ultimately, with the experience of kind of living on a cancer ward and seeing, sadly, the death of many children, including the death of my own daughter, I was really determined to kind of work in the field of actually drugging these targets that we were working so hard to discover in genomics. So I chose Exscientia. It's a really fantastic company. And what appealed to me about it was their mission to kind of encode and then automate drug discovery in cancer, in small molecule cancer therapeutics, because I think they've taken a really pragmatic, a kind of non-theoretical path to actually making drugs quickly, efficiently, but also effective drugs that really will make an impact on the patient community that I've learned to kind of care so much about.
Thank you very much, Parker. That's a very powerful story. Our next guest is Margo Georgiadis, who is the co-founder and CEO of Montai Health. It's another fantastic company that is utilizing AI to develop medicines for chronic diseases. Margo, can you tell us a little bit more about your background and also the work that you do at Montai Health?
Well, thanks, Charlotte. It's great to be here with Parker and Guillermo. Actually, somewhat similarly to Parker, I actually started my career as a technologist, spent 3 decades focused on how technology can transform companies and industries, and had the privilege of working across multiple industries. And I spent nearly a decade at Google, scaling the innovative platforms into tens of billions. I ran the Americas, North and South America, as well as building all the translational engineering teams that help take products to market. And then I had the opportunity to apply this leading edge technology as a CEO of several companies, most recently a company called Ancestry, which is really the combination of those experiences at Google and Ancestry led me to seeing this tremendous untapped opportunity. You know, when we think back to the early development of AI and the transformational benefits of things like image recognition to pathology, radiology, and neurology, or then the increasing complex data sets that enabled us at Ancestry to better understand risk predictions for disease, understanding family health history, NGS screening. How could we think really transformationally about designing clinical trials? And similar to Parker, I was really drawn to the area of chronic disease due to firsthand motivation of seeing family members that were suffering from these debilitating diseases to which there was really no cure, but there was potentially an opportunity for understanding pathways that could enable us to modulate them. Yet most of the most powerful therapies were biologics, which were accessible to less than 5% of the patients, and even the most severe patients, less than 50% had access. How could we think fundamentally differently about the chemistry that we might start with and the technology we could use that would enable us to create transformational therapies for these chronic diseases, in many cases replacements of the biologics, which is what we're focusing on first at Montai, as well as trying to drug difficult targets like transcription factors that are really important in the regulation of diseases, particularly immunoregulations. And, you know, for me as a career technologist who really has thought about how do we use AI and use it to translate or reimagine industries. You know, life science is such an important area for us to tackle, not just because of the massive human impact, but also the fact that, you know, we're investing on average $1 billion with a 10% odds of success, and that's just not good enough for the patients that need to be served. And I had the opportunity to build this company with Flagship Pioneering, which is really known for being willing to take leaps to reimagine how things are done and take a long-term view, which when you're trying to build one of these platform companies, you have to be willing for other people, you know, to challenge you and say, well, that's just not possible. Or, you know, other people have tried that before. Why are you doing that? And in our case at Montai, I think that's very true, right? I get asked all the time. We decided to focus on aggregating the world's largest understanding of human co-evolved chemistry. So this is chemistry that would have existed in our food, herbs, and traditional medicines. And our fundamental hypothesis was that in nature's power of coevolution was a wide variety of diverse structures that could engage any biology. And what we needed to do is be able to harness the power of that and stop fishing in the same enamine library pond that we've been fishing on for many, many decades, but create models that enabled us to understand diverse chemistry and how to turn it into great drugs with efficiency. And that's really what we've built at Montai. We've combined sort of AI that's enabled us to curate this massive understanding of this diverse chemistry, which you could only efficiently aggregate computationally, because otherwise it would cost billions, right, just to synthesize all these molecules. But computation enables us to do this for the first time, and then using AI to precision match it into biological pathways, and for the first time build a stacking knowledge of the intersection between chemistry and biology, and completely unconstrained chemistry and biology. And it took us several years to build the platform, but we have demonstrated that in this power of coevolution, we can find solutions to the most important biological pathways. And we're really excited about the opportunity to advance these therapeutics where at least we have some human built in, which is a scaffold that we know has been in the human body. Of course, dose makes the poison at the end of the day, and we'll need to medchem most of this chemistry to make it a great therapeutic. But at the same time, I'd rather start with a provenance of knowing it's been in a body for millennia versus I have absolutely no signal.
At last, I'd like to welcome Guillermo de Angel, Executive Director and Rare Disease Therapy Head at AstraZeneca's Center for Genomics Research. Guillermo, could you tell us a little bit more about your background and your work at AstraZeneca?
Absolutely, Charlotte, and I'm very, very happy to be here. And I always say that I have a very odd background because I came to the pharmaceutical company through the technology side, so it's great to actually be joined by both Parker and Margo, who actually have this parallel journey. I originally trained as an electrical engineer and then as an applied mathematician, and I always tell semi-seriously that I was doing data science before data science was called data science, and I was doing machine learning before actually machine learning was fashionable. And that's actually very, very true. I mean, you know, I became interested in these subjects back in the late 90s as a grad student. And it was an interesting time because there was a little bit of a lull in the progress of data science, which again, I mean, the term didn't even exist then, and machine learning was a very niche application. But I was trying to do that, and I had set myself to actually do software development and as a career, which is what I initially started to do after I got my PhD. But then in a parallel way as to Parker, two personal things happened that made me reassess my path and how I was going to focus my career. And you know, in my case, it was my mother passing away from cancer, and then my uncle, who I was very close with, developing a rare genetic disease later in life. And then that actually made me think, okay, how do I actually use the toolbox that I have acquired in applied math, software development, machine learning into actually problems that matter, than actually that have impact in healthcare, and how could I eventually perhaps save lives, or at least make lives better using the toolbox that I had developed. So I retrained in computational biology and bioinformatics, and eventually I landed at the Broad Institute back in 2010 as a computational biologist. So this was a time when next generation sequencing was really being developed and really started to be applied at scale to actually get insights into the population. So we went into the era of not just sequencing one person at a time, but sequencing actually different cohorts of people at a time. So I actually had the pleasure of being involved with these early stage population genetics initiatives like the 1,000 Genomes Project, and then later on what would become the Exome Aggregation Consortium later, and really starting to scale the way we saw genome sequencing. So and then we started to develop AI and machine learning approaches to actually, first of all, clean up the data and figure out what was a false positive in the genomic sequences versus what was a true positive, and actually then start to apply the insights from there into really population genetics insights, and then later into clinical genetics. So after the Broad Institute, I went to McKinsey to work in their business technology office as a technology consultant, which was a very, very good learning opportunity for me, and thinking about how companies and organizations develop their strategy, how companies develop their technologies, and how actually we could use some of the scientific insights in other areas to create impact for organizations. And then in 2015, I joined what was then Alexion Pharmaceuticals in their data science, genomics, and bioinformatics team. So there, I started to apply all of these different tools that I had developed in the toolbox specifically for rare disease genetics. So that's when actually, for example, I started to collaborate with Genomics England, where I later on met Parker, in trying to understand how do we actually apply genetics, machine learning, in general AI, to actually get insights into disease. And rare diseases having such a strong genetic component were a very, very natural place where actually we could use these insights. So we ended up applying quite a bit of novel machine learning approaches on identifying patients, and actually this is work that actually directly led in the Genomics England cohort to identifying 16 previously unidentified patients, which was a very, very nice story, because in rare diseases, one of the biggest hurdles that we have is actually the patient diagnosis. It's very, very well known, and it's actually a sad reality that in many cases it takes years and years for patients to be correctly identified and diagnosed. So we found actually that we could use genetic information, that we could actually develop machine learning approaches to actually speed up that rare disease odyssey in patients. And actually my team that in Alexion did quite a bit of work on actually how to rapidly identify using genetics and machine learning rare disease patients. And this is actually an effort that is in many ways still ongoing. Then back in 2020, just before the pandemic, I took leadership of the bioinformatics and data science team in Alexion, which was a great opportunity for me to actually scale up and apply all of the insights that I had been learning sort of like at a organization-wide setting. And then in 2021, AstraZeneca bought Alexion, and Alexion became the rare disease unit in AstraZeneca. And last year, I took my current role as a rare disease therapy area head at the Center for Genomics Research in AstraZeneca. Now, the Center for Genomics Research is a company-wide group that serves all therapeutic areas, not just rare disease, but actually all throughout AstraZeneca. And our bold ambition is to transform the way we do healthcare and the way AstraZeneca develops drugs using genomics information. And our goal is eventually to have up to 2 million sequences at our disposal in 2026, and we're well on our way to actually achieve that number. But we don't see that number just as a number that is to be checked off. And we actually want to obviously maximize the impact in healthcare and in drug development, so that this data provides. And we have been able, with this data, with this volume of data, to actually develop novel ways, for example, in which we can use proteomics and genomics combined with clinical measurements to actually, for example, predict biomarkers for different diseases. Or actually, for example, we can actually do very powerful association tests between different phenotypes and different genetic loss of function models. For example, one thing that we're very interested now is in finding essentially protective variants, so essentially genetic variants that protect us against disease, because that can actually provide novel insights into how to develop therapeutics, right? Essentially, using what we call nature's clinical trials and using the genetic variation in the population to actually give us insights into novel targets for diseases of interest.
Great, thank you so much. So let's dive into our topic. I'll start with my first question. Where do you expect to see the biggest impact of AI on drug development? Is it more at the drug identification stage of the process, of all the way downstream to clinical trial stratification, or somewhere in between? I'd love to hear your perspectives on this.
So I can jump in if nobody else wants to. So I think that it's going to be all throughout the drug discovery process. And we have been able to see, for example, great insights into target identification. So for example, how do we use machine learning combined with genetic information to actually point us to, for example, targets that we hadn't thought about before. And it could be, for example, not just linking novel mechanisms of disease with particular genes, but also, for example, as I was mentioning before, in protective signals. For example, in how can we use, for example, AI with genetics to find variations, natural variations that protect us against disease, and then how do we actually develop therapeutics based on that insight. And then downstream in the pipeline, we have been seeing great examples for in the way, for example, that we design drugs, that we were using generative AI and some of the same tools that are now in vogue with ChatGPT and these large language models, repurposing that same toolbox, for example, in generating novel proteins or novel small molecules that actually speed up the drug discovery process.
Yeah, I think just building on that, all of these are going to be transformed, as data access and models continue to improve. I think for all of us, it's humbling because human biology is so hugely complex and there's still so much that we don't know. It's interesting in the literature, and a lot of the public opinion, right? Many people are really saying, wow, where's this promise? We've been talking about AI for a while, and where's the outcome? And I think what's really challenging is that when you do it for target discovery, right, the time from target discovery and validation all the way to getting a therapy in the clinic and approved, it's a really long time frame. And so I think we're not going to see in the near term just huge numbers of these molecules, right, getting in from the target discovery side. I think Where we can make faster progress is in areas like clinical trials, where we really can think differently about patient stratification. And we're spending so much of our dollars in that area that the ability to do that with greater efficiency, I think, can be transformational in the economic model and enable greater investment in other areas if we can raise the bar for success, right? The fact that in a small molecule development, on average, success rates in the clinic are 7, and we consider, right, antibodies at 12 to be heroically better, right, just sends us a signal that there's enormous opportunity, right, for us to think differently about the whole chain. I think for me personally, I think the areas that we'll see the greatest productivity outside of clinical trials is going to be in this precision matching of chemistry and biology, and being able to get to therapies that are more precise, that do have are engaging the pathways in ways that reduce off-target issues and better take into account a more integrated pathway cascade, is where we're going to be able to, really drive improvements. And I think that's where this network learning in terms of pathway biology, in terms of getting a much richer set of multimodal data signals, is enabling us to understand with much better fidelity than we've ever had before, building on some of Guillermo's comments, our ability to create that level of precision on these pathways against the areas that we know that we've then validated to be important in the disease. And so I think it's just an incredibly exciting time. To me, it's like the tip of the iceberg as you think about the transformation of other technology categories that I think each of us has been part of. You find this new technology, people see all these amazing things you can do with it. It tends to be a feature in the beginning where people will take existing ways of doing things and optimize them. Then the most exciting piece is when we can start with the patient outcome that we want to get to, and then we reverse engineer the entire process. We turn it from an artisanal into an engineering problem where we can iterate much more with much greater fidelity, much more greater efficiency to get to that outcome faster. And I think for all three of us, just listening to those introductions, I think that's what we're all obsessed with, is how do we improve outcomes for patients? That's why we started this, because it was a passion for us. And that's, for me, being involved in this as somebody who comes at this with an AI first mentality, but also it's about the beauties and the blend. It's about the art and the science coming together and enabling us to have unprecedented insights and optionality throughout this process to speed it up and improve outcomes.
Yeah, I'd love to build on that answer as well. I think you've put a very complimentary panel together. So just to put it in the context of what Exscientia is up to, I mean, we are a company that focuses on cell-intrinsic oncology in the small molecule space. And we're very much an AI native company, much like Margo's. And we're a full stack company. So very much as Margo just said, we start with the patient. We kind of sometimes call it reverse translation. So we put a lot of work into collecting primary patient tissue, which tells us not just about target biology beyond kind of mice, we call it sometimes from mice to men, but also about kind of translational pathways and biomarkers that can be used to stratify clinical trials in a more meaningful way. We also work in the chemistry space. So we're very much working on the automation of the design process to try to in silico design better drugs. And then we've gone further than that by actually building in Oxford a fully automated chemistry studio, so that as well as designing in silico drugs, we can then manufacture them on site and accelerate that learning loop. And that's a really, really critical part of this whole exercise, because the world of oncology in particular has not just enormous efficiency problems, but also enormous efficacy problems in its kind of traditional manifestation in large pharma. So to kind of put that in numbers, from an efficiency perspective, it can take 8 to 10 years to bring a small molecule to market, and even more than that in many cases. And estimates, we used to talk about oncology costing around 2.4 billion to get a drug to market. I think recent estimates from last year are more like 6.7 billion, which is absolutely extraordinary. And then even when drugs get to market, there tends to be around a 4% success rate in clinical trials. So there's not only an enormous cost and kind of time lag here, but there's an effectiveness challenge as well. Now, both of those can be addressed by AI. So to put that into perspective, when you're designing compounds to be tested in mice and other animal models, usually large pharma companies, the kind of industry standard is to make between 5 and 10,000 compounds before you bring those to kind of lead optimization to be tested in animals. We are aiming to massively triage that effort and really only build around 200 compounds because we've pre-screened for synthesizability, for toxicity, and we only build compounds that we have high confidence will actually work and can be made. So that drives enormous kind of efficiencies from a cost perspective and a time perspective. But the real beauty is not just what it does for efficiency. That allows us to kind of go around this loop from the dry lab to the wet lab and back again many more times, because instead of taking 4 to 5 or 6 months to do that, which is the typical process for biotechs that work with CROs, we can close a learning loop in 3 to 4 weeks. So we can iterate that loop, optimizing towards a target product profile in weeks rather than months. And that allows us to kind of gradually iterate it again and again. And I think that's the real key about AI here. It's not about having the best performing model for protein ligand binding, or the best physics model for free energy calculations, or the best synthesizability model. It's really, as I think Margo said before, an engineering challenge of stringing all of those models together in the optimal way and having real intimacy between your kind of in silico design and then your manufacturing, automating, and testing. That brings not just efficiency, but also effectiveness to this field.
Thank you. So you've talked a little bit about the range of applications that you see AI having in the field, and you touched upon some of the constraints within which you're operating to choose what to focus on. I wanted to talk a little bit about data. To what extent data availability and scale is influencing the areas you're focusing into, and are there areas right now that you'd be really interested in looking at but don't necessarily have the data that you require to look at?
Do you want me to jump off with that? So data availability is a fascinating kind of challenge in this space. To give a perspective from the chemistry space, some of the very large kind of chemical libraries out there have 10 to the 9 or even 10 to the 10 chemicals in them. There are kind of various estimates of the chemical search space. The Lipinski space is sometimes described as 10 to the 23. Other people have described the kind of chemical search space is 10 to the 63. So whichever way you go, there are kind of 10 orders of magnitude between the kind of availability of data in libraries today and the potential chemicals out there that could be transformed into drugs. So what we know, what that tells us is that this is not a training exercise. This is an AI learning exercise. What you often need to do is start from a kind of chemical start point, which can come from these libraries, which can come from wonderful new discoveries like AlphaFold, or increasingly it can come from kind of proprietary data that, in our case, we generate in our automated studio in Oxford. But the important thing is that from that chemical start point, you have to learn your way into the area of chemical space that is going to really deliver a result, a druggable, an affordable, a safe, and an efficacious drug. So that's really where generative AI comes in. It's a learning problem, and that's where we're putting a lot of our energy today at Exscientia.
Yeah, absolutely. And to build upon that, especially in the area that I work on, which is in rare diseases, and specifically rare disease genomics, data availability has always been an issue, right? I mean, because a lot of the times the volume of patients that are out there for one particular disease might be in the few hundreds or even less sometimes. So we have to be really, really smart about how to get the data, how to use it, Sometimes there is only positive training data, but no negative training data. So we have to be smart. Okay, how do you balance the data sets, right? I mean, how do you actually build reliable learning models to actually explore that space that you want to explore in? And this is somewhere where, for example, one of the things that Parker was mentioning about exploring more of the search space really comes at handy, right? And that's also where, for example, the recent push into generative AI has shown to be so valuable and has really so much potential, right? And because it essentially allows us to explore that space that we cannot get into by just sampling the current data points that we have, because those might be very, very sparse.
Yeah, just to reinforce that, I think for all of us, the concept of time from idea to experiment is critical, and we can control this much better with automation than we could before. But building on Parker's point, I do think that there's sufficient data now to, in terms of generating high throughput screening, large data sets, and the power of computational tools is enabling us to scale these things at a pace and sophistication that just wasn't possible even a few years ago. I know that the technology that underpins a lot of the work we do at Montai in terms of scaled graph deep learning, right? We just didn't even have the computational processing capacity to understand the number of iterations to go into as we do. So 87% of our scaffolds don't exist in those Enamine libraries that Parker was talking about. And so many of the models were trained really scaffold up to understand, right, how these different things could be iterated. In our case, we built a fundamental capability that's scaffold agnostic. And so we've essentially built a superhuman chemist that understands how to read the language of chemistry and how it connects to our biology, but it can do it within any scaffold. And I think for each of us, we're trying to push a boundary. And for us, that was the boundary we wanted to push, because the perception was these structures are too complex. They're very difficult to prosecute. They might have hard, difficult off-target effects, et cetera. So how do we actually do a better job of understanding the richness that's sort of hidden in plain sight and use AI to unearth that in a new and a different way? And what we've been able to do by using AI is to create that understanding. So that connection between aggregating that chemistry and the biology is what is enabling us to build that stacking understanding of that fidelity. And also by building deep network pathway maps between chemistry and biology, right? Many pathways are counter screens for the other pathways. So when you stack this knowledge over time, your ability to drive to precision and more richly understand what is actually happening in those pathways is so important. Because at the end of the day, the most important signal, which is the human signal, is always the sparsest, right? I think for all of us, if every pharma and biotech in the world were to share every piece of clinical trial data down to the patient level for every trial, what worked and didn't, and we were able to share that, our ability to advance the state of the art in this industry would go far faster. Unfortunately, that isn't possible, versus when we think about all human language, right, has been aggregated. And that's why those LLMs in the English language have been able to advance, so much more quickly. And so I think it really challenges us, the people that are in this space, to think differently about how do we find other creative ways to get to ground truth human signals, which is, for example, one of the reasons why we focused in on this diverse co-evolved chemistry to look for untapped solutions. And it shouldn't be surprising that somebody would do this using computation, because this is how drug development started. In fact, if you look at many of the most powerful drugs like artemisinin or progesterone or statins, or many of these things all came directly from nature. Most of the new PPIs, glues, degraders, all these things were inspired by what people saw in nature and then turned it into an executable modality. And I think what we really need to do is really be able to understand these things broadly. What has nature created holistically? Start with the biology, not the technology, and let the biology pull the chemistry that will best enable it to modulate as precisely as possible. And I think, these are the types of problems that we can now solve using AI that just were not doable, even just a few years ago. So I think it's a really exciting time.
Thank you, Margo. That leads on to my next question. Many AI models are black boxes, and it's sometimes difficult compared to conventional approach to understand exactly how we got to the results that we got to. So in your respective works, do you need to develop AI model where you need to understand the why and have more control over the parameters? Or are you actually happy with testing things empirically a little bit more quickly? When do you feel like you need to bring more traditional methods of validation, maybe to validate the assumptions that you make in terms of what you're reading from the results? Or is it simply okay as well to accept that the underlying mechanisms might remain inscrutable?
Yeah, let me jump off of that one. I think this topic of explainability is more often discussed in the kind of clinical space, where you have to explain mechanism of action to a doctor at the very least, and often to patients. So I can understand why you might think this is less important to drug discovery, but actually that is not the case at all, and it all relates to this principle that I think we've all agreed on, that this is a learning problem. If you're using AI just as a filter to tell you which scaffold to hop to next, but you don't know why, then you're not learning, and you're not encroaching further into this kind of chemical search space that I think all three of us have referred to. So, for example, the models we have built, I like this scaffold agnostic term that Margo mentioned. We also built abilities to kind of to hop between our rings and other scaffolds in the kind of core of the molecule, and we have kind of auto rendering tools that will actually colorize the compounds, the molecules, and show which piece of the molecule at an atomistic level is responsible for potency, or is responsible for kind of reversibility or selectivity, for example. And being able to really understand which part of the scaffold you therefore want to keep and which one you want to kind of move away from or iterate around is absolutely vital, and really helps kind of bring humans into the loop, which is very much the way that we work. We are not yet, to be clear, in case any of the listeners are confused, we're not at all yet in the space where you can put a target product profile and some atomy requirements into a kind of search engine, and at the end a fully rendered molecule pops out the other end. Maybe in 30 years we'll get there, but at the moment there is still some kind of human interpretation required to triage in this kind of long cascade down to ultimately a lead candidate.
Yeah, I so agree with that. I'm so glad you said that. I think in almost every industry, right, that's been transformed, the ability to really transform the type of decisions you can make as early as possible with the greatest amount of information possible is really what enables acceleration, right? You can reimagine the process. And so, I think similar to Parker, I think we built our platform to supercharge drug developers. It is about the AI and the artisans coming together. It's just they get to do it in a totally new way that enables both to have unprecedented insights and optionality to make better decisions that are more likely to get to great outcomes. So, AI is fundamental, but, at the end of the day, we can't get away from expert judgment. And I think the key for us has been creating, I think similar to the example Parker gave, proprietary tools that actually help to understand what AI is actually doing at each step, because it's at the edges that you find the insight. And I think that's something we did in many other, if you think about every single category that you've seen massive transformation in, you have to be able to provide those sorts of tools so people can touch it, feel it, understand it, discuss it, because then we're actually not advancing the learning. We're not thinking about the pieces, because what goes into AI has to be first curated. So the quality of the data set massively impacts, right, what comes out the other side. And I think most of us don't talk enough about the enormous efforts that we go to standardize, curate, quality control, to ensure that those fundamental data sets, right, don't have bias, or if they do, we understand what the implications of that are. And I think we see that a lot, right, in the discussions, especially when you get to generative AI, of what can happen, right, as it loops on itself, right? So you have to avoid the hallucinations. But one of the things I think, really works well for us is, even at the earliest possible stage, right, you have that target candidate profile, even when your understanding hits, we are getting that early to those top 200 candidates where we actually know not only that they can potently specifically engage the biology, we have our ability to understand that precisely, but we can also understand all their other drug-like properties so that you understand exactly what you're signing up for as early as possible. And what that enables is much more efficient decision-making at each step in the process. And I think this is a huge leap for an industry that historically was an artisanal set of handoffs, step by step. Or even if you start at the earliest stage in terms of understanding, pathway precision, for us, we can integrate with AI something that would be impossible to do in a wet lab. We're able to integrate multiple orthogonal assays at the same time in the computationally to get to that precision. And I think this is such a wow for scientists because they would previously have to make trade-offs. That reductive, right, view actually does not lead you to the same conclusions. And so that's what's so exciting. But their ability to see and peel those layers back and understand what chemistry survives and doesn't as you look at that, and what are the similarities and differences, and giving them those tools actually inspires them to make better decisions, to advance at every step the most promising chemistry. And, the biggest challenge in this industry is the cost, right, on the wet lab or the clinic, right, is really huge. So we have to use AI to enable that optionality so we don't fall in love with the scaffold, right? We have to have enough options that people actually follow the data and the science. And intuition is great, but intuition gets confused when you only have one option. And, as I've worked with a lot of the best-in-class drug developers, that's what I'm seeing is really driving transformational improvement, is that when you have those options, you will then constantly scrutinize and ensure, that you are advancing only the best possible candidate.
Absolutely. And to build upon that, one thing that we have been debating is to what extent do we actually want to invest resources in providing that explainability? And always the conclusion has been, yes, we need to. And for every single model that we produce, we need to actually be able to articulate what are the features driving the predictions, because that's a way also to cement confidence in its use by essentially the non-AI scientists in the organization, and providing sort of like that set of key features that are driving things is also a way to validate biologically what's going on, because sometimes depending on the training data, if you have subtle biases, the model might go into some strange directions, and the ability for you to then probe what is really driving the predictions is key to actually as an internal QC measure, right? I mean, which we think is very important. And we have seen that over and over, especially when you have all of these, potentially noisy, potentially biased input data sets, where we really need to be mindful of what are the predictions, essentially, why are the predictions being made the way they are, right? And again, even if eventually the models validate themselves, we actually need to cement confidence downstream of us. So I think that's also key.
Thank you. I want to close out with your perspective on an area of AI or drug development that may not be on everyone's radar, but that you think will be impactful. What do you have in mind? What are maybe the more niche applications of AI in the context of drug discovery?
That's a great question, and maybe I can hop in very quickly. And if you see where the biggest need is, I think that, as was mentioned before, we need to do something about the rate of success of our candidates going not just into the clinic, but into approved drugs. And the industry as a whole has a little bit of an abysmal record, especially in terms of efficacy. And if you see where most of the failures are, which are the more expensive failures, are late, either phase 2 or phase 3, where drugs actually fail to actually show efficacy. And that's where the failures hurt the most, right? I mean, because running such a clinical trial is very, very, very expensive, right? And to some extent, you have to pay not just for the trials that succeed, but actually you also have to pay for the trials that fail, right? And you have all of these accumulated costs that you need to recoup as a drug development organization. So I don't think it's a niche application, but it's actually where there is the more need on actually how do we actually empower better success rates in clinical trials. And I think this is going to be a combination of finding the right targets and the right safety, but also the right patients and really, much better matching of patients with particular drugs and much better use of knowledge of the biology, which will require us to be more creative in the way we think about the matching between patients and drugs and diseases. I think that's actually where the highest potential for impact that will have.
I think just building on that, I think there's a lot of opportunities, over the next, as we stack this network knowledge, both at the patient side, but also in the interactions between chemistry and biology, to get to scalable personalization in a way that we have not before. And I think, we know that we go through these, whether it's a chronic disease or cancer, right? We end up with these step progression of the different things that we offer patients, but the non-responder rates are still really high. And we have to find a way to really understand as precisely as we can. And I think with the higher and higher fidelity genetic screening, blood screening, right, et cetera, we have the ability to actually understand a person's dysregulation and be able to understand not just single pathway, but synergistic pathway solutions. And I think we've seen a lot of progress on this in cancer, right? We've certainly seen it in several of the rare diseases. And I think, we have to be able to really treat the person ultimately in both the prosecution of medicine and make it easier for clinicians, but also in the development, of therapies. I think, this is something that, is really possible, as we, scale our knowledge, in these areas. I think what's really tricky for me as someone who's come into life sciences later in my career is because this industry mainly earns its economic returns from an asset. The data, if you think about other industries where the chips or the things that really create the foundational insights have a way to earn an economic return. And so that incentive then to build and stack that knowledge, right, can gain a return level that enables those advances at a more rapid pace. And there's much more data sharing, right? People are collaborating with each other throughout the ecosystem in a much more integrated way to drive that stacking learning. And, I think when you add that component of the economic framework and the regulatory framework together, that just does create, some barriers to advancement. And I think it's really up to us as, leaders in this industry to find more and more new and creative ways to collaborate with each other, because at the end of the day, almost all of us got into this because we cared about the patient outcome. And I think if we as an industry could really challenge ourselves to say, how do we all think about, the ways in which we could collaborate, to accelerate progress for everyone? I think it would be a really fantastic outcome for all.
Yeah, I'd agree with all of that. And maybe if I could end by saying something a little bit controversial. So I really, again, liked how Margo brought this concept of kind of bringing the artisanal and the kind of, I don't know what you call it, the scalable processes of drug discovery together. And that it is true that there are some artisanal pieces. It's hard to imagine how they will scale up. So, for example, testing in mice and then mini pigs. If there are people out there working out how you can run GLP tox in a mouse more quickly than we do today, please contact us, because we'd love to know that. That's not a part of the process we're working on. We consider that still an artisanal exercise. But there are the part of the process which really can speed up is this learning loop. And as I've tried to describe it, it's this loop that traverses from in silico molecular design through to kind of automation and test, not just of the molecule, but also of the assays that are going to that the molecules need to be tested against, the biochemical assays. And so the controversial thing I want to say is I don't actually think this is largely an AI problem. AI is already going fast, and it is already speeding up incredibly quickly. In fact, I mean, the rate of the pace that now AI can handle large quantities of data really is putting the choke point on the data availability, not the machine learning, the computation itself. But I think what ultimately will be the rate limiting step is the lab automation, not the design studio. Because if we have to wait months and months and months to go through synthesis steps to synthesize thousands and thousands of compounds and then assay them, and it takes months and months to build assay arrays, then that is really going to be a kind of choke point on this highly scalable design, make, test part of our process. So I think what we will see and what we need and what Exscientia is investing very heavily in is really the scaling up and the automation of the lab itself, of moving chemicals through make and purification and verification very quickly, through building tens, hundreds of assays per month, through having a kind of fully automated lab really that can operate 24 hours a day at night when there are no people there, and really be the flywheel that accelerates that learning loop and really kind of brings this intimacy between the chemistry and the computation. So I don't think you can AI your way to making a drug. We still, I think, for the foreseeable future, actually have to make things and test them in assays and then ultimately in animals. I would say, therefore, it's not AI, it is the automation, which is the choke point in that scalable piece. And I'm excited to see how we and also other players in the industry are going to really change that in the coming years.
Thank you, Parker. I think it's a really good insight for us to close our discussion today. Thank you very much. It's been a highlight to hear your different perspectives on the potential that AI is bringing to the future of medicine, and also to see how your respective works are already contributing to this progress. It's hugely exciting to me, and I think will be to our listeners, to see the many applications that AI is already having and will continue to have in the future in this field. So thank you very much for your contributions. It's been a really good discussion.
Thank you.
Thank you.
My pleasure.